Hello students, today we will learn about genetic engineering of plants and its methodology. It is the second video of this part. Let's begin. First is organization. I teached half about organization of diplasmid. Again starting from organization of diplasmid. So the diplasmid exist as independent replicating circular DNA molecules within the agrobacterium cells. The tDNA is variable in length in the range of 12 to 24 kilobase pair, which depends on the bacterial strain from which diplasmid come. A strain named nopalin of diplasmid have one tDNA with length of 20 kilobase kb, while octopine strains have two tDNA regions referred to as TL and TR that are respectively 14 kb and 7 kb in length. The diplasmid has three important regions. First is tDNA, second is virulence region, third is open catabolism region. Now from starting let's know about tDNA region. This region has has the genes for the biosynthesis of auxin, cytokinin and opin and is flanked by left and right borders. These three genes, OX, Cyto, and OX, are referred to as oncogenes as they are the determinants of the tumor phenotype. Now, tDNA borders. A set of 24 kilobase sequences present on the either side of tDNA are also transferred to the plant cells. It is now clearly established that the right border is more critical for tDNA transfer and tumorigenesis. Now let's know about the second region. Second region is virulence region. The genes responsible for the transfer of tDNA into the host plant are located outside tDNA and the region is referred to as Vir or virulence region. Vir region codes for the protein involved in tDNA transfer. At least 9 Vir gene operands have been identified. These include Vir A, Vir G, Vir B1, Vir C1, Vir D1, D2 and D4 and Vir E1, Vir E2. Now, third region is open catabolism region. This region codes for the proteins involved in the uptake and metabolisms of opins. Besides these three, there is Ori region that is responsible for the origin of DNA replication, which permits the DI plasmid to be stably maintained in A tumescence. tDNA transfer and integration. Let's know about tDNA transfer and integration. The process of tDNA transfer and its integration into the host plant genome are three. Let's know about this. First point is signal induction to agrobacterium. The wounded plant cells release certain chemicals, phenolic compounds and sugars which are recognized as signals by agrobacterium. The signals induced result in a sequence of biochemical events in agrobacterium that ultimately helps in the transfer of tDNA of T. Second is attachment of agrobacterium to plant cells. The agrobacterium attaches to plant cells, though polysaccharides, particularly cellulose fibers, produced by the bacterium. Several chromosomal virulence genes responsible for the attachment of bacterial cells to plant cells have been identified. Production of virulence proteins. As the signal induction occurs In the agrobacterium cells attached to the plant cells, a series of events take place that results in the production of virulence proteins. To start with, signal induction by phenolic stimulates Vir-A, which in turn activates Vir-G. This induces expression of virulence genes of Thai plasmids to produce the corresponding virulence proteins, that is D1, D2, E2B, etc. Certain sugars like glucose, galactose, xylose that induce virulence genes have been identified. Now, let's know about the production of tDNA strand. The right and left borders of tDNA are organized by Vir D1, Vir D2 proteins. These proteins are involved in the production signal stranded tDNA, its protection and export to plant cells. The single stranded tDNA gets attached to Vir D2. Now let's know about transfer of tDNA out of agrobacterium. The single-stranded tDNA Vir-G2 complex in association with Vir-G is exported from the bacterial cell. Vir-B products from the transport apparatus. Next is, let's know about the sixth point, that is transfer of tDNA into plant cells and integration. The tDNA Vir-D2 complex crosses the plant plasma membrane. In the plant cells, tDNA gets covered with Vir E2. This covering protects the tDNA from degradation by nucleases. Vir D2 and Vir E2 interact with a variety of plant proteins which influences tDNA transport and integration. The tDNA Vir D2 Vir E2 plant protein complex enters the nucleus through nuclear pore complex. Within the nucleus, the tDNA gets integrated into the plant chromosome through a process referred to illegitimate recombination. This is different from the homologous recombination as it does not depend on the sequence similarity. Now let's know about the hairy root disease of rhizogenes R1 plasmids. Agrobacterium rhizogenes can also infect plants, but this results in hairy roots and not crown gulls, as is the case with 8 Agrobacterium tumefaciens. The plasmids of Agrobacterium rhizogenes have been isolated and characterized. This plasmid referred to as RI plasmids, that is RI stands for root inducing plasmids, are of different types. Some of the RI plasmid strains possess genes that are homologous to TI plasmid. Example, auxin biosynthetic genes. Instead of virulence genes, RI plasmids contain a series of open reading frames on the tDNA. The products of these genes are involved in the metabolism of plant growth regulators, which get sensitized to auxin and leads to root formation. Now let's know about the vectors of Agrobacterium rhizogenes. As it is done with Agrobacterium tumefaciens, vectors can be constructed by using Agrobacterium rhizogenes. These vectors are alternate strategies for gene transfer. However, employment of Agrobacterium rhizogenes based vectors for plant transformation is not common, since more efficient systems of Agrobacterium tumefaciens have been developed. Now, let's know about the importance of hairy roots. Hairy roots can be cultured in vitro and thus are important in plant biotechnology. Hairy root systems are useful for the production of secondary metabolites, particularly pharmaceutical proteins. Now, let's know about the Ti plasmid derived vector system. The ability of Ti plasmid of Agrobacterium to genetically transfer plants has been described. It is possible to insert a desired DNA sequence into the tDNA region and then use Agrobacterium tumefaciens to deliver these genes into the genome of plant cell. In this process, TI plasmids serve as natural vectors. However, there are several limitations to use TI plasmids directly as cloning vectors. TI plasmids are large in size, that is 200 to 800 kilobase. Smaller vectors are preferred for recombinant experiments. For this region, large segments of DNA of TI plasmid not essential for cloning must be removed. Absence of unique restriction enzyme sites on TI plasmids. The phytohormones, that is auxin and cytokinin, produced to prevent the plant cells being regenerated into plants. Therefore, auxin and cytokinin genes must be removed. Now, opin production is transferred plant cells, lower the plant yield. Therefore, the opin synthesizing genes which are of no use to plants should be removed. TI plasmids cannot replicate in E. coli. This limits their utility as E. coli is widely used in recombination experiment. As an alternate arrangement to is to add an origin of replication to TI plasmid that allows the plasmid to replicate in E. coli. Considering these limitations, TI plasmids based vectors with suitable modifications have been constructed. These vectors are composed of some components. Let's know about the components. The right border sequence of tDNA, which is absolutely required for tDNA integration into plant cell DNA, a multiple cloning site that promotes the insertion of cloned gene into the region between tDNA borders. An origin of DNA replication that allows the plasmid to multiply in E. coli. A selectable marker gene, that is neomycin phosphotransferase, for appropriate selection of the transformed cells. Two types of TI plasmid derived vectors are used for genetic transformation of plants: co-integrate vectors and binary vectors. First, let's know about co-integrate vector. In the co-integrate vector system, the disarmed and modified TI plasmid combines with an intermediate cloning vector to produce a recombinant TI plasmid. Now let's know about the production of a disarmed TI plasmid. The tDNA genes for the hormone biosynthesis are removed, that means disarmed. In the place of the deleted DNA, a bacterial plasmid, that is PBR322 DNA sequence is incorporated. This disarmed plasmid, also referred to as receptor plasmid, has the basic structure of tDNA necessary to transfer the plant cells. Construction of intermediate vector. The intermediate vector is constructed with some components. Let's know about these components. The PBR322 sequence, DNA homologous to that found in the receptor TI plasmid. The a plant transformation marker, that is PTM, example a gene coding for neomycin phosphotransferase 2, in short NPT2. This gene confers resistance to kanamycin in the plant cells and thus permits their isolation. A bacterial resistant marker, example a gene coding for spectinomycin resistance. This gene confers spectinomycin resistance to recipient bacterial cells and thus permits their selective isolation. A multiple cloning site where foreign genes can be inserted. A co or E1 region of replication which allows the replication of plasmid in E. coli but not in Agrobacterium. An ORI-T sequence with basis of mobilization site for the transfer of intermediate vector from E. coli to Agrobacterium. Production and use of co-integrate vectors. The desired foreign gene is first cloned in the multiple cloning site of the intermediate vector. The cloning process is carried out in E. coli, the bacterium where the cloning is most efficient. The intermediate vector is mated with agrobacterium so that the foreign gene is mobilized into the latter. The transferred agrobacterium cells with receptor Ti plasmid and intermediate vector are selectively isolated when ground on a minimal medium containing spectromycin. The selection process becomes easy since E. coli does not grow on a minimal medium in which agrobacterium grows. Within the agrobacterium cells, intermediate plasmid gets integrated into the receptor Ti plasmid to produce co-integrate plasmid. This plasmid containing plant transformation marker example NPT2 gene and cloned target gene between tDNA borders is transferred to plant cells. The transferred plant cells can be selected on a medium containing kanamycin when the plant and agrobacterium cells are incubated together. Now let's know about the advantages of co-integrate vector. Target genes can be easily cloned. The plasmid is relatively small with a number of restriction sites. Intermediate plasmid is covalently cloned in E. coli and transferred to Agrobacterium. Now let's know about the binary vector. The binary vector system consists of an Agrobacterium strain along with a disarmed Ti plasmid called Vir helper plasmid. That is, the entire tDNA region, including borders, deleted while Vir genes is retained. It may be noted that the both of them are not physically linked or interlinked. A binary vector with tDNA can be replicated in E. coli and Agrobacterium. Let's know about some components of this the binary vector. First point is left and right borders that delimit the tDNA region. Second is the plant transformation marker PTM example NPT2 that confers kanamycin resistance in plant transformed cells. A multiple cloning site for introducing target or foreign genes. Next point is a bacterial resistant marker that is tetracycline resistant gene for selecting binary vector cloning is in E. coli and Agrobacterium. Fifth point is ORI-T sequence for conjugal mobilization of the binary vector from E. coli to Agrobacterium. A broad host range origin of replication such as RK2 that allows the replication of binary vector in Agrobacterium. Production and use of binary vector. Let's know about the production and use of binary vectors. The target gene of interest is inserted into the multiple cloning site of the binary vector. In this way, the target gene is placed between the right and left border repeats and cloned in E. coli. By a matching process, the binary vector is mobilized from E. coli to Agrobacterium. Now, the virulence gene protein of tDNA facilitate the transfer of tDNA of the vector into plant cells. Now, let's know about the advantages of binary vectors. The binary vector the binary vector system involves only the transfer of a binary plasmid to agrobacterium without any integration. This is in contrast to co-integrate vector system wherein the intermediate vector is transferred and integrated with disarmed Ty plasmid. Due to convenience, binary vectors are more frequently used than co-integrate vectors. Now let's know about the plant transformation techniques in agrobacterium. Agrobacterium mediated technique is the most widely used for the transformation of plants and regeneration of transgenic plants. The important requirements for gene transfer in higher plants through agrobacterium mediated. Now we'll discuss. The explants of the plant must produce phenolic compound for activation of virulent genes. Transformed cells or tissues should be capable to regenerate into whole plants. In general, most of the agrobacterium mediated plant transformation have been has some basic protocols. Let's know about the protocols. First is development of agrobacterium carrying the co-integrate or binary vector with the desired gene. Next point is identification of a suitable explant. Example cells, protoplasts, tissues, callus, and organs. Third point is co-culture of explants with agrobacterium. Fourth is killing of agrobacterium with a suitable antibiotic without harming the plant tissue. Fifth point is selection of transformed plant cells. Sixth is regeneration of whole plants. Now let's know about the advantages of agrobacterium mediated transformation. This is a natural method of gene transfer. Next point, agrobacterium can conveniently infect any explant, example cells, tissues or organs. Even large fragments of DNA can be efficiently transferred. Stability of transferred DNA is reasonably good. Transformed plants can be regenerated effectively. Now let's know about the limitation of agrobacterium mediated transformation. There is a limitation of host plants for agrobacterium since many crop plants are not infected by it. In recent years, virulent strains of agrobacterium that can infect a wide range of plants have been developed. The cells that regenerate more efficiently are often difficult to transfer. Example, embryonic cells lie in deep layers which are not easy targets for agrobacterium. Now let's know about the virus mediated gene transfer or plant viruses as vectors. Plant viruses are considered as efficient gene transfer agents as they can infect the intact plants and amplify the transferred genes through viral genome replication. Viruses are natural vectors for genetic engineering. They can introduce the desirable gene into almost all the plant cells since the viral infections are mostly systemic. Plant viruses are non-integrative vectors. The plant viruses do not integrate into the host genome in contrast to the vectors based on tDNA of Agrobacterium tumefaciens which are integrative. The viral genomes are suitably modified by introducing desired foreign genes. These recombinant viruses are transferred, multiplied, and expressed in plant cells. They spread systematically within the host plant where the new genetic material is expressed. Now let's know about the criteria for a plant virus vector. An ideal plant virus for its effective use in gene transfer is expected to possess some characteristics. Let's know about the characteristics. The virus must be capable of spreading from cell to cell through plasmodesmata. Next point is the virus genome should be able to replicate in the absence of viral coat protein and spread from cell to cell. This is desirable since the insertion of foreign DNA will make the viral genome too big to be packed. The recombinant viral genome must elicit little or no disease symptoms in the infected plants. The virus should have a broad host range. Next point is the virus with DNA genome is preferred since the genetic manipulations involve plant DNA. The three groups of viruses Colimoviruses, Gemini viruses, and RNA viruses that are used are vectors for gene transfer in plants. Plants we'll discuss now. Colimoviruses as vectors. The colimoviruses contain circular double-stranded DNA and are spherical in shape. Colimovirus are widely distributed and are responsible for a number of economically important diseases in various crops. The colimovirus group has around 15 viruses, and among these, cauliflower mosaic virus is the most important for gene transfer. The other colimoviruses include carnation yeast virus, dahlia mosaic virus, mirabilis mosaic virus, strawberry vein binding viruses. Now let's know about the cauliflower mosaic virus. CAMB. CAMB infects many plants, members of Crucifera datura, and can be easily transmitted even mechanically. Another attractive feature of CAMB is that the infection is systemic and large quantities of viruses are found in infected cells. The genome of CAMB consists of a 8 kb, that means 8024 base pair, relaxed but tightly packed circular DNA with 6 major and 2 minor coding regions. The genes 2 and 7 are not essential for viral infection. Use of CAMB in gene transfer. For appropriate transmission of CAMB, the foreign DNA must be encapsulated in viral protein. Further, the newly inserted foreign DNA must not interfere with the native assembly of the virus. CAMB genome does not contain any non-coding regions wherein foreign DNA can be inserted. It is fortunate that two genes, namely gene 2 and gene 7, have no essential functions for the virus. It is therefore possible to replace one of them and insert the desired foreign gene. Gene 2 of CAMB has been successfully replaced with a bacterial gene encoding dihydrofolate reductase that provides resistance to methotrexate. When the chimeric CAMB was transmitted to turnip plants, they were systematically infected and the plants developed resistance to methotre- methotrexate. Now let's know about the limitations of CAMB as a vector. CAMB vector has a limited capacity for insertion of foreign genes. Infective capacity of CAMB is lost if more than a few hundred nucleotides are introduced. Helper virus cannot be used since the foreign DNA gets expelled and wild type viruses are produced. Let's know about the Gemini viruses as vectors. The Gemini viruses are so named because they have geminate morphological particles, that is twin and paired capsid structures. These viruses are characterized by possessing one or two single stranded circular DNAs. One replications, that is a single stranded DNA from an intermediate double stranded DNA. The Gemini virus can infect a wide range of crop plants, monocotyledons and dicotyledons, which attract plant biotechnologists to employ these viruses for gene transfer. Curly top virus, that is CTB, and maize stick virus MSB, and bean golden mosaic virus BGMB are among the important Gemini viruses. It has been observed that a large number of replicative forms of a Gemini virus genome accumulate inside the nuclei of infected cells. The single stranded genomic DNA replicates in the nucleus to form a double stranded intermediate. Gemini virus vectors can be used to deliver, amplify, and express foreign genes in several plants or explants. However, the serious drawback in employing Gemini viruses as vectors is that it is very difficult to introduce purified viral DNA into the plants. An alternate arrangement is to take the help of agrobacterium and carry out gene transfer. RNA plant viruses as vectors. Let's know about RNA plant viruses as vector. There are mainly two types of single stranded RNA viruses. First is monopartite virus and second is multipartite viruses. Let's know about the monopartite viruses. These viruses are usually large and contain divided genomes for all the genetic information. Example, tobacco mosaic virus. Now let's know about the multipartite viruses. The genome in these viruses is divided into small RNAs which may be in the same particle or different particle. Example, Brom mosaic virus BMV. HMV contains 4 RNAs divided into between three particles. Plant viruses in general are characterized by high level of gene expression, good efficiency to infect cells and spread to different tissues. But the major limitation to use them as vector is the difficulty of joining RNA molecules in vitro. Now let's know about the use of cDNA for gene transfer. Complementary DNA copies of RNA viruses are prepared in vitro. The cDNA so generated can be used as a vector for gene transfer in plants. This approach is tedious and cumbersome. However, some success has been reported. A gene sequence encoding chloramphenicol resistant has been inserted into bromosaic virus genome. This genome expression, however, has been confined to protoplast. Now let's know about the limitations of viral vectors in gene transfer. The ultimate objective of gene transfer is to transmit the desired genes to subsequent gene generations. With virus vectors, this is not possible unless the virus is seed transmitted. However, in case of vegetative propagated plants, transmission of desired traits can be done. Example, potatoes. Even in these plants there is always a risk for the transfer of transferred genes to be lost anytime. For the reason referred now uh, we discussed, plant biotechnologists prefer to insert desired genes of interest into a plant chromosome. Now let's know about the direct or vectorless DNA transfer. The term direct or vectorless transfer of DNA is used when the foreign DNA is directly introduced into the plant genome. Direct DNA transfer methods rely on the delivery of naked DNA into the plant cells. This is in contrast to the agrobacterium or vector mediated DNA transfer, which may be regarded as indirect methods. Majority of the direct DNA transfer methods is simply ineffective, and in fact several transgenic plants have been developed by this approach. Now let's know about the limitations of direct DNA transfer. The major disadvantage of direct gene transfer is that the frequency of transgene rearrangements is high. This results in the higher transgene copy number and high frequencies of gene silencing. Now let's know about the types of direct gene transfer. The direct transfer can be broadly divided into three categories. First is physical transfer methods, that is electroporation, particle bombardment, microinjection, liposome fusion, silicon carbide fibers. Second is chemical gene transfer method, that is polyethylene glycol PEG, in short PEG, mediated diethyl aminoethyl, that is DEAE, dextran mediated calcium phosphate participation. Third is DNA ambition DNA imbibition by cells, tissues, organs. Now let's know about the physical gene transfer methods. First is electroporation. Electroporation basically involves the use of high field strength electrical impulses to reversibly permeabilized the cell membranes the for the uptake of DNA. This technique can be used for the delivery of DNA into intact plant cells and protoplasts. The plant material is incubated in a buffer solution containing the desired foreign or target DNA and subjected to high voltage electrical impulses. This results in the formation of pores in the plasma membrane through which DNA enters and gets integrated into the host cell genome. In the early years, only protoplasts were used for gene transfer by electroporation. Nowadays, intact cells, callus culture, and immature embryos can be used with suitable pre and post electroporation treatments. Electroporation has been successfully used for the production of transgenic plants of many cereals, example rice, wheat, maize. Now let's know about the advantages of electroporation. This technique is simple, convenient, and rapid besides being cost effective. The transformed cells are at the same physiological state after electroporation. Next point is efficiency of transformation can be improved by optimizing the electrical field strength and addition of spermidine. Limitations of electroporation. Let's know about the limitations of electroporation. Under normal conditions, the amount of DNA delivered into plant cells is very low. Efficiency of electroporation is highly variable depending on the plant material and the treatment conditions. Degeneration of plants is not very easy, particularly when protoplasts are used. Now let's know about the particle bombardment, or in short listics. Particle bombardment is the most effective method for gene transfer and creation of transgenic plants. This method is versatile due to the fact that can be successfully used for DNA transfer in mammalian cells and microorganisms. The microprojectile bombardment method was initially named as biolistics by its inventor Sanford in 1988. Biolistics is a combination of biological and ballistics. There are other names of this technique, that is particle gun, gene gun, bioblaster, etc. Microcarriers, that is microprojectiles, the tungsten or gold particles coated with DNA are carried by microcarriers. These microcarriers are inserted into the apparatus and pushed downward by rupturing the disc. The stopping plate does not permit the movement of microcarrier, while the microcarriers are propelled at the high speed into the plant material. Here the DNA segments are released which enter the plant cells and integrate with the genome. Plant material used in bombardment. Let's know about the plant materials which are used in bombardment. There are two types of plant tissues that are commonly used for particle bombardment. First is primary explants, second is proliferating embryonic tissues. Let's know brief about these points. Primary explants which can be subjected to bombardment that are subjectively induced to become embryonic and regenerate. Second is proliferating embryonic tissues that can be bombarded in cultures and then allowed to proliferate and regenerate. In order to protect plant tissues from being damaged by bombardment, cultures are maintained on high osmotic media or subjected to limited plasmolysis. Let's know about the transgene integration in bombardment. It is believed that the gene transfer in particle is a two-stage process. In the pre-integration phase, the vector DNA molecules are spliced together. This results in fragments carrying multiple gene copies. Now let's know about the integrative phase. Integrative phase is characterized by the insertion of gene copies into the host plant genome. The integrative phase facilitates further transgene integration, which may occur at the same point or a point close to it. The net result is that particle bombardment is frequently associated with high copy number at a single locus. This type of single locus may be beneficial for regeneration of plants. Now let's know about the success of bombardment. The particle bombardment technique was first introduced in 1987. It has been successfully used for the transformation of many cereals. Example: rice, wheat, maize. In fact, the first commercial genetically modified crops, such as maize containing Bt toxin gene, were developed by this approach. Now, let's know about the factors affecting bombardment. Several attempts are made to study the various factors and optimize the system of particle bombardment for its most efficient use. Some of the important parameters we will discuss now. Nature of micro Nature of microparticles. Inserts metals such as tungsten, gold, platinum are used as microparticles to carry DNA. These particles with relatively higher mass will have a better chance to move fast when bombarded and penetrate the tissues. Now, let's know about the nature of tissues or cells. The target cells that are capable of undergoing division are suitable for transformation. Now, let's know about the amount of DNA. How much amount of DNA will use? The transformation may be low when too little DNA is used. On the other hand, too much DNA may result in high copy number and rearrangement of transgenes. Therefore, the quantity of DNA used should be balanced. Recently, some workers have started using the chemical aminosiloxane to coat the microparticles with low quantities of DNA adequate enough to achieve high efficiency of transformation. Environmental parameters. Let's know about the environmental parameters. Many environmental variables are known to influence particles bombardment. These factors like temperature, humidity, photo period, etc., influence the physiology of the plant material and consequently the gene transfer. It is also observed that some explants after bombardment may require special regimes of light, humidity, and temperature, etc. The technology of particle bombardment has been improved in recent years, particularly with regard to the use of equipment. A commercially produced particle bombardment apparatus, namely PDS 1000 by HC, is widely used these days. Now, let's know about the advantages of particle bombardment. First point is gene transfer can be efficiently done in organized tissues. Second point is different species of plants can be used to develop transgenic plants. Now let's know about the disadvantages of particle bombardment. The major complication is the production of high transgene copy number. This may result in instability of transgene expression due to gene silencing. Next point is the target tissue may often get damaged due to lack of control of bombardment velocity. Next point is sometimes undesirable chimeric plants may be regenerated. Now let's know about the microinjection. Microinjection is a direct physical method involving the mechanical insertion of the desired DNA into a target cell. The target cell may be the one identified from intact cells, protoplast, callus, embryos, meristems, etc. Microinjection is used for the transfer of cellular organelles and for the manipulation of chromosomes. The technique of microinjection involves the transfer of the gene through a micropipette, that is zero point 0.5 to 10 micrometer tip, into the cytoplasm or nucleus of a plant cell or protoplast. While the gene transfer is done, the recipient cells are kept immobilized in agarose embedding and held by a suction holding pipette. As the process of microinjection is complete, the transformed cell is cultured and grown to develop into a transgenic plant. In fact, transgenic tobacco and Brassica napus have been developed by this approach. The major limitations of microinjection are that it is slow, expensive, and has to be performed by trained and skilled personnel. Now let's know about the liposome mediated transformation. Liposomes are artificially created liposome vesicles containing a phospholipid membrane. They are successfully used in mammalian cells for the delivery of proteins, drugs, etc. Liposomes carrying genes can be employed to fuse with protoplast and transfer the genes. The efficiency of transformation increases when the process is carried out in conjunction with polyethylene glycol (PEG). Liposome mediated transformation involves adhesion of liposomes to the protoplast surface. Fusion fusion at the site of attachment and release of plasmids inside the cell. Now let's know about the advantages of liposome fusion. Being present in an encapsulated form of liposomes, DNA is protected from environmental insults and damage. DNA is stable and can be stored for some time in liposome prior to transfer. Application to a wide range of plant cells. There is a good reproducibility in this technique. Now let's know about the disadvantages of liposome fusion. The major problem with liposome mediated transformation is the difficulty associated with the regeneration of plants from transferred protoplast. Silicon carbide fiber mediated transformation. The silicon carbide fibers, in short SCF, are about 0.3 to 0.6 micrometer in diameter and 10 to 100 micrometer in length. These fibers are capable of penetrating the cell wall, the plasma membrane, and thus can deliver DNA into the cells. The DNA coated silicon carbide fibers are vortexed with plant material. During the mixing, DNA adhering to the fiber enters the cells and gets stably integrated with the host genome. The silicon carbide fibers with the trade name whiskers are available in the market. Now let's know about the advantages of SCF mediated transformation. First is direct delivery of DNA into intact walls cells. This avoids the protoplast isolation. Procedure is simple and does not involve costly equipments. Now let's know about the disadvantages. Silicon carbide fibers are car- uh, can, car- carcinogenic and therefore have to be carefully handled. Now, second point is the embryonic plant cells are hard and compact and are resistant to SCF penetration. In recent years, some improvements have been made in SCF mediated transformation. This has helped in the transformation of rice, wheat, maize, and barley by using this technique. Let's know about the chemical gene transfer methods. Polyethylene glycol mediated transformation, PEG, in short PEG. Polyethylene glycol, polyethylene glycol in the presence of divalent cations using Ca2 plus destabilizes the plasma membrane of protoplast and renders it permeability permeable to naked DNA. In this way, the DNA enters nucleus of the protoplast and gets integrated with the genome. The procedure involves the isolation of protoplast and their suspension addition of plasmid DNA followed by a slow addition of 40% PEG 4000 W by V dissolved in mannitol and calcium nitrate solution. As this mixture is incubated, protoplast get transformed. Now let's know about the advantages of PEG mediated transformation. A large number of protoplasts can be simultaneously transformed. This technique can be successfully used for a wide range of plant species. Now let's know about the disadvantage. The DNA is susceptible for degradation and rearrangement. Random integration of foreign DNA into genome may result in undesirable traits. Regeneration of plants from transformed protoplast is a difficult task. DEA dextran mediated transfer. The desirable DNA can be complexed with a high molecular weight polymer diethyl aminoethyl dextran and transferred. The major limitation of this approach is that it does not yield stable transformants. Now let's know about calcium phosphate co-precipitation mediated transfer. The DNA is allowed to mix with calcium chloride solution and isotonic phosphate buffer to form DNA calcium phosphate precipitate. When the actively dividing cells in culture are exposed to this precipitation for several hours, the cells get transformed. The success of this method is dependent on the high concentration of DNA and protection of the complex precipitate. Addition of dimethyl sulfoxide increases the efficiency of transformation. Let's know about the DNA imbibition of cells or tissues. Some workers have seriously tried to transform cells by incubating cell suspensions, tissues, embryos, and even seeds with DNA. The belief is that the DNA gets imbibed and the cells get transformed. DNA inhibition approach has met with little or no success. Now completed. Next video will continue from marker genes for plant transformation. Any questions regarding this, you can ask me in the comment box. I'll definitely answer the questions and subscribe my channel. Thank you.